Most of the world didn't have it until colonization started in the 15th century.
And it was discovered that the rhesus factor could be positive or negative. Most humans in the world are rhesus positive.
There is something hidden inside the veins of nearly half the people walking this earth right now. It is not a disease. It is not a defect. In fact, for most of human history, it may have been the single most important reason their ancestors survived at all. Scientists call it blood type O. For decades, medicine treated it as nothing more than a clinical label, a convenient classification used to match donors to patients in emergency rooms. But a growing body of research, published in some of the most respected scientific journals in the world, is telling a completely different story. What researchers have uncovered about type O blood goes far beyond transfusions and donor compatibility. It touches the deep mechanics of immunity, the hidden forces that shaped human evolution, the catastrophes that nearly erased entire populations, and a biological resilience that may have quietly decided who survived and who did not across thousands of years of human history. This is that story. To understand why type O blood matters so much, you first need to understand what it actually is, and what it is not. Human blood is classified into four main groups: A, B, AB, and O. The difference between them comes down to tiny molecular structures on the surface of red blood cells called antigens. People with type A blood have A antigens on their cells. Those with type B have B antigens. People with AB carry both. But type O is defined entirely by absence. Type O individuals carry neither A nor B antigens on the surface of their red blood cells. Instead, their immune systems produce antibodies against both A and B antigens, a natural defensive posture against anything carrying those molecular signatures. This absence is precisely why type O blood became known as the universal donor. When type O blood enters a body carrying a different blood type, the recipient's immune system has little to react against. There are no A or B antigens to identify as foreign. It is biologically neutral in that regard, and in a trauma bay where there is no time for blood typing, that neutrality has saved countless lives. But here is where the story takes a turn. That same molecular neutrality, that absence of surface antigens, has consequences that reach far beyond blood transfusions. It changes how pathogens interact with the body. It influences how the immune system responds. It affects how blood clots, how inflammation unfolds, and how the body weathers diseases that have killed millions across history. Scientists have been piecing these connections together for decades, and what they have found is quietly staggering. The molecular landscape of type O blood is not simply an empty canvas. It is an active biological state with downstream consequences that ripple through nearly every major system in the human body. And to truly grasp the depth of what scientists have discovered, you have to begin with one of the oldest and deadliest killers in human history. Of all the diseases that have shaped human genetics, malaria stands near the top. Plasmodium falciparum, the most deadly strain of the malaria parasite, has been killing humans for so long and in such extraordinary numbers that it has left permanent marks on the human genome. The prevalence of sickle cell trait in sub-Saharan Africa is one famous example, a genetic mutation that causes painful health problems in some circumstances but offers protection against malaria in others. Evolution had, in a brutal sense, struck a bargain. Blood type O appears to be part of a similar bargain, and the mechanism was clarified in a landmark study published in the Proceedings of the National Academy of Sciences. Researchers studying children in Mali, West Africa, found something remarkable. Blood group O was associated with a 66% reduction in the odds of developing severe malaria compared to all non-O blood groups combined. The odds ratio was 0.34, and the statistical confidence was extremely high. This was not a marginal finding. This was a decisive protective effect playing out in real children facing one of the world's most dangerous diseases. The biological mechanism involves a process called rosetting. When malaria parasites infect red blood cells, those infected cells become sticky. They begin to clump together with healthy red blood cells, forming clusters called rosettes. These clusters can block small blood vessels, particularly in the brain and kidneys, leading to the severe organ failure and coma that defines the deadliest forms of malaria. Researchers found that the A and B antigens on the surface of non-O red blood cells appear to facilitate this clumping. The antigens act almost like biological glue, helping the infected cells adhere to healthy ones. Type O cells, lacking these antigens, are far less likely to form these deadly rosettes. The infected cells simply cannot grip them as effectively. Further research published in Nature Medicine confirmed that type O alleles have been evolutionarily selected for in populations where malaria has historically been most intense. Scientists examining the rifin proteins produced by the malaria parasite found that these proteins preferentially bind to A and B blood group antigens. Type O individuals, lacking those antigens, essentially deny the parasite one of its most effective weapons. In malaria-endemic regions across sub-Saharan Africa, type O blood frequencies range from 40 to 80% of the population. In some South American regions, where malaria has also been a persistent killer, the frequency climbs even higher. This is not coincidence. This is natural selection visible in real time, written in the distribution of a blood type across continents. Here is where the story becomes genuinely complex, and where researchers encountered one of the most striking paradoxes in the entire field of blood type research. If type O blood offers meaningful protection against malaria, what about cholera? The answer is deeply counterintuitive, and it reveals that the relationship between blood type and disease is far more nuanced than a simple advantage or disadvantage. The link between cholera and blood type O was first identified by Barua and Paguio in 1977, and it has been confirmed by numerous investigators since. The finding, published and reviewed in the American Journal of Tropical Medicine and Hygiene, is striking in its severity. When cholera swept through Peru in 1991, a large-scale epidemic that shocked global health authorities, researchers studying the outbreak found that individuals with blood type O were 8 times more likely to be hospitalized with the most severe forms of the disease. Not twice as likely. Not 3 times. 8 times. The mechanism appears to involve the cholera toxin's interaction with blood group antigens. Researchers found that the cholera toxin binds to the blood group O determinant in multiple orientations and with greater affinity than it binds to blood group A antigens. In simpler terms, the very absence of A and B antigens seems to expose type O individuals to a more powerful form of the toxin's assault on the intestinal lining. The gut cells of type O individuals mount a stronger cellular response to the cholera toxin, a cascade that drives the catastrophic fluid loss that makes cholera so deadly. This is a biological double-edge that science is still trying to fully understand. In some confrontations with disease, the absence of A and B antigens is a shield. In others, it is a vulnerability. The disease landscape of any given region, across any given era of history, could determine whether type O was an asset or a liability. In an environment where malaria was the dominant killer, type O may have conferred survival advantages that outweighed everything else. In a region devastated by cholera, the calculation would flip entirely. This paradox helps explain why blood type distributions across the world are not uniform. Evolution is not selecting for type O universally. It is selecting for the genetic profile best suited to the specific disease pressures of each environment, each era, each population's particular history of epidemics and outbreaks. Beyond infectious disease, type O blood carries a distinct signature in the cardiovascular system, and this one tends to work consistently in its favor. The key involves a protein called von Willebrand factor, a clotting agent that plays a central role in blood coagulation. Research published across multiple peer-reviewed journals, including studies drawing on large population cohorts, has established that individuals with blood type O have, on average, approximately 25% lower activity of von Willebrand factor in their plasma compared to individuals with types A, B, or AB. This matters enormously. Von Willebrand factor helps blood clot when injury occurs, which is essential for healing. But too much von Willebrand factor activity increases the risk of dangerous clot formation inside blood vessels, the kind that causes heart attacks and strokes. People with blood type A tend to have higher levels of this clotting protein. A meta-analysis involving over 145,000 cardiovascular cases found that blood groups AB had the strongest statistical association with ischemic stroke and myocardial infarction compared to other blood groups. Blood group O showed the lowest cardiovascular risk profile in these comparisons. A large Dutch study looking at individuals with von Willebrand disease, a condition characterized by deficiency of this very clotting protein, found something revealing. These individuals experienced 39% fewer arterial thrombotic events than reference populations, along with significantly lower rates of ischemic stroke and coronary heart disease. The pattern maps almost directly onto what scientists observe in type O blood carriers: lower von Willebrand factor, lower clotting risk, lower cardiovascular events over a lifetime. A 2022 study published in Neurology found that the difference in early-onset stroke risk based on blood type was especially pronounced in people under the age of 60. In younger adults, before the accumulated damage of high blood pressure and diabetes begins to dominate cardiovascular risk, blood type itself emerged as a meaningful independent factor. This is not to say that type O blood makes someone immune to heart disease. It does not. Lifestyle, diet, genetics beyond the ABO locus, and countless other factors all play roles. But at the population level, across thousands of individuals studied over years, type O blood appears to carry a measurable and consistent cardiovascular advantage rooted in the biology of clotting. In 2020, as the SARS-CoV-2 pandemic swept the globe and researchers scrambled to understand why some people became severely ill while others experienced only mild symptoms, blood type entered the picture again. A study published in Annals of Internal Medicine, drawing on population-level cohort data, found that type O individuals had a lower risk of testing positive for COVID-19. Another study published in the journal Blood found that blood group A enhanced SARS-CoV-2 infection. Research from the UK Biobank, analyzed and published in Arteriosclerosis, Thrombosis, and Vascular Biology in 2024, confirmed that people with blood type O were less likely to have tested positive during the pandemic's first wave and also showed a lower risk of cardiovascular complications following COVID infection. The proposed mechanism centers on the immune biology of type O carriers. People with type O blood naturally produce antibodies against both A and B antigens. Researchers found that these anti-A antibodies can bind to A-like antigens expressed on the spike protein of the SARS-CoV-2 virus. This binding appears to block the virus's interaction with the ACE2 receptor, the primary gateway through which the coronavirus enters human cells. In essence, the very antibodies that type O individuals produce as a routine feature of their blood type may partially neutralize the virus before it can establish a foothold. An Italian study published in the American Journal of Hematology estimated the relative risk of SARS-CoV-2 infection in type O subjects at 0.74 compared to other blood groups, a meaningful reduction in risk. It is important to note that type O blood does not make anyone immune to COVID-19. People with type O can contract the virus, experience severe illness, and transmit it to others. But at the population level, the biological interaction between type O antigens, type O antibodies, and the virus appears to create a modest but real protective effect. All of these modern findings gain an even stranger dimension when you look back into human history and ask a deceptively simple question. Why is type O blood so extraordinarily common in indigenous populations of the Americas? In many indigenous communities across the continent, blood type O does not merely dominate, it is nearly the exclusive blood type present. Studies of certain Amazonian tribes have found type O frequencies approaching 100%. Among indigenous communities across Central and South America, the near-total absence of A and B blood types is a documented scientific reality, not an exaggeration. The traditional scientific explanation rests on two mechanisms, the founder effect and genetic drift. The founder effect refers to the loss of genetic variation that occurs when a new population is established by a small number of individuals. The original migrants who crossed the Bering Land Bridge from Siberia into the Americas tens of thousands of years ago represented a relatively small founding group. If that group happened to carry a high proportion of O alleles, their descendants would reflect that skew. Research published in Blood Group O alleles in Native Americans, drawing on analysis of 1,873 X chromosomes across Native American and other continental populations, confirmed that a major population bottleneck occurred during the peopling of the Americas. The loss of specific haplotypes frequent in Eurasia, combined with the rise in frequency of a third haplotype rare elsewhere, indicated that the founding population was small and genetically constrained. But additional research has complicated this clean founder effect narrative. A study published in the American Journal of Physical Anthropology by researchers Halverson and Balnick used ancient DNA to test whether the high frequency of type O in modern Native Americans reflected the original founding population's genetics, or whether it represented a subsequent change. The results pointed toward a more complex picture, one in which historical events after the initial peopling, particularly the catastrophic population collapse following European contact and the disease pandemics of the 1500s and 1600s, may have played a significant role in reshaping blood type distributions. If the diseases brought by Europeans disproportionately killed individuals with A and B blood types, perhaps due to immune interactions that made them more vulnerable to certain pathogens, then survivors would have been disproportionately type O. The surviving gene pool would not reflect the original founding population as much as it would reflect the survivors of a biological catastrophe. This hypothesis remains actively debated among geneticists and anthropologists. What is not debated is the extraordinary degree of type O concentration in the Americas, or the fact that this concentration reflects some combination of ancient bottlenecks, evolutionary selection pressures, and possibly post-contact disease dynamics that science is still working to untangle. What ties all of these pieces together, the malaria protection, the cardiovascular differences, the COVID-19 associations, the ancient population patterns, is the fundamental biological reality of what type O blood actually represents at the molecular level. Type O is defined by the absence of A and B antigens. But that absence is not a simple emptiness. It is an active biological state with downstream consequences for how the immune system recognizes and responds to threats. The ABO blood group gene produces enzymes that either add or fail to add specific sugar molecules to the surface of red blood cells. In type A individuals, an enzyme adds a molecule called N-acetyl D-galactosamine to the precursor H antigen, creating the A antigen. In type B individuals, a different enzyme adds D-galactose, creating the B antigen. In type O individuals, the ABO gene carries mutations, typically a single nucleotide deletion in exon 6, that create a non-functional enzyme. No sugar is added. The H antigen remains exposed. This H antigen, expressed abundantly on type O red blood cells, interacts with the world differently than A and B antigens do. It presents a different surface to pathogens, to immune cells, to clotting factors, and to inflammatory signals. The full scope of what this means for human health is still being investigated, but the accumulating evidence suggests that the molecular landscape of type O blood has consequences across multiple biological systems simultaneously. What scientists are increasingly recognizing is that the ABO blood group gene does not merely determine what is on the surface of red blood cells. It is associated with downstream effects on angiotensin-converting enzyme activity, red blood cell count, hemoglobin concentration, von Willebrand factor levels, factor VIII levels, platelet function, and inflammatory markers. It is, in a sense, a regulatory hub whose effects ripple through the body in ways researchers are still mapping. And the more they map it, the more they find that this single genetic locus, inherited from both parents, expressed on every red blood cell in the body, touches human biology in ways that were simply not visible when blood typing was first developed a little over a century ago. It would be tempting, at this point, to conclude that type O blood is simply superior, that nature has selected for it and the data proves it is the winner in the genetic lottery. But that conclusion would be wrong, and science does not support it. Type O individuals face a well-documented elevated risk of peptic ulcers. The bacterium Helicobacter pylori, which causes the majority of stomach ulcers, binds more effectively to the H antigen expressed abundantly on the gastric lining of type O individuals. Research has consistently found that type O people are more susceptible to H. pylori infection and the ulcers it causes. As the cholera data shows, type O individuals face dramatically higher severity of cholera disease once infected. In environments where cholera is endemic and treatment scarce, being type O could be a life-threatening vulnerability rather than a survival advantage. Research on certain cancers has also shown complexity, with some studies suggesting that the absence of A and B antigens on cancer cells may in specific contexts reduce the immune system's ability to recognize and destroy them. The picture that emerges is not one of a superior blood type, but of a blood type with a distinct biological profile, one that provides significant advantages against some of history's most destructive diseases while carrying vulnerabilities against others. The evolutionary fitness of type O blood has always depended on which diseases were present in the environment, and in many of the environments where human populations face the most intense selective pressure from infectious disease, the malaria belts of Africa, the pathogen-rich environments of tropical South America, type O's protective effects against severe illness appear to have been the dominant force. The high frequency of type O we observe today in those regions is the cumulative outcome of that dominance, playing out across hundreds of generations of births, deaths, epidemics, and survivals. The science of blood type O, assembled from decades of peer-reviewed research across multiple fields, reveals something genuinely remarkable, a biological signature that has been actively shaped by the worst diseases humanity has ever faced. Type O blood is more than a donor category. It is a document. Written in the language of molecular biology, it records millions of years of encounters between human immune systems and the pathogens that tried to kill them. The high frequency of type O blood in malaria-endemic regions of Africa reflects the selection pressure of a parasite that has been killing humans since long before written history began. The extraordinary prevalence of type O in indigenous populations of the Americas reflects the complex history of migration, isolation, population bottlenecks, and the catastrophic disease waves that reshaped entire continents. The lower von Willebrand factor levels in type O individuals, giving them their cardiovascular advantages, may reflect selection pressures operating over thousands of generations. The partial protection against COVID-19 infection, mediated by antibodies that cross-react with viral surface proteins, is the latest chapter in a very long story about how the immune landscape encoded in blood type shapes survival odds. None of this makes any blood type better or worse as a human being. It does not mean type O individuals are healthier overall, or that people with type A or B are at a disadvantage in modern life. Modern medicine, sanitation, and nutrition have largely decoupled our health from the brutal selective pressures that shaped blood type distributions in ancient populations. But the scientific story of type O blood is a window into something profound: the fact that the human body carries the memory of its own past crises. The distribution of blood types across the globe is a map of ancient epidemics, migrations, and survival trials. The frequencies we observe today are the cumulative outcome of who lived and who died across thousands of generations of encounters with disease. The researchers who have built this picture, geneticists, immunologists, epidemiologists, and evolutionary biologists working across continents and decades, have collectively revealed that what looks like a simple medical classification is actually one of the most fascinating stories in human biology. Type O blood is common. It is often treated as ordinary. But the science says it carries something extraordinary inside it: the quiet record of survival itself.